If bacteria from a depressed person's gut can change how a mouse responds to reward and motivation, the uncomfortable question is whether our own gut ecosystem is silently influencing the brain circuits that regulate mood, drive, and the ability to think clearly. New research published in Molecular Psychiatry does not stop at correlation. It identifies specific synapses, specific dopamine pathways, and specific structural changes in the brain that follow when gut microbiota from bipolar depression patients are transplanted into animal models.
Welcome to Dr. Mercola's Cellular Wisdom. Stay informed with quick, easy-to-listen summaries of our latest articles, perfect for when you're on the go. No reading required. Subscribe for free at mercola.com for the latest health insights.
Hello and welcome to Dr. Mercola Cellular Wisdom. Bipolar disorder affects roughly 37 million people worldwide and involves recurring cycles of depression and periods of elevated or irritable mood. Dr. Mercola highlights that for decades, this has been understood through a purely brain-centered lens focused on neurotransmitters and neural circuits. But the growing body of evidence points toward a gut-brain interaction where immune signals, stress hormones, and microbial metabolites all contribute to how mood and cognition are regulated. This study tested that directly by asking whether gut dysbiosis could influence the brain networks involved in depressive symptoms.
The researchers focused on two brain structures. The medial prefrontal cortex plays a central role in decision-making, emotional regulation, and evaluating rewards and consequences. The ventral tegmental area is a key dopamine hub. Communication between these two regions supports motivation, reward processing, and emotional drive. Disruptions in this pathway align directly with depressive symptoms like low initiative and reduced ability to experience pleasure. Rather than simply correlating gut bacteria with symptoms, the researchers collected gut microbiota from people experiencing bipolar depressive episodes and transplanted it into mice. The animals developed depression-like behaviors, including reduced movement and less interest in rewarding treats.
The structural findings were equally significant. The mice showed decreased dendritic spine density in the medial prefrontal cortex. Dr. Mercola explains that dendritic spines are like docking ports for incoming signals. Fewer spines means fewer functional contact points between neurons, which translates into weaker communication within circuits, regulating emotion and decision-making. The study also identified disruptions in local protein production at the synapse, a process critical for synaptic plasticity. When synapses cannot remodel properly, the brain loses some of its capacity to adapt and maintain cognitive resilience.
At the circuit level, the analysis revealed fewer connections between ventral tegmental area inputs and prefrontal cortex glutamate neurons. Alongside that, reduced connectivity came a measurable decrease in dopamine response. The reward and drive circuit looked blunted, creating a clear biological bridge between dysbiosis and features like low motivation and diminished ability to feel pleasure. This moves the conversation beyond abstract associations between gut health and mood into territory where specific neural mechanisms are being altered by microbial signals.
The article broadens to a diagnostic issue as well. Bipolar depression and major depressive disorder can look clinically similar, which means misdiagnosis happens frequently. Dr. Mercola highlights research examining whether gut microbiome profiles might help distinguish between the two conditions. Bipolar disorder was associated with reduced alpha diversity compared to healthy controls, suggesting a less resilient gut ecosystem. Across both conditions, a shared finding emerged. Beneficial short-chain fatty acid producers were depleted. Short-chain fatty acids, especially butyrate, are produced when gut bacteria ferment dietary fiber. Butyrate supports the intestinal lining, helps regulate immune balance, and plays an important role in gut-brain communication.
In untreated bipolar disorder specifically, studies reported lower abundance of butyrate-producing genera including Roseburia, Faecalibacterium, and Coprococcus. Dr. Mercola emphasizes that losing these populations weakens a critical biological support system for both gut barrier integrity and brain signaling. The cognitive dimension is also important because bipolar disorder frequently includes cognitive impairment that persists even between mood episodes. A separate study in BMC medicine found that bipolar patients with clear cognitive impairment had a significantly different gut microbiome profile compared to those without cognitive issues. Genera involved in glucose metabolism correlated more with cognitive performance, while inflammation-associated bacteria tracked more closely with depressive severity.
What makes this especially compelling is that supplementation with microbiota from healthy donors partially restored function in animal models. The mice showed improved emotional behavior, enhanced cognitive performance, and recovery of neuronal plasticity markers. Dr. Mercola presents that as meaningful support for the biological relevance of microbial balance in shaping both mood and higher-order thinking.
The broader framework describes several pathways through which gut dysbiosis reaches the brain. When the gut barrier is compromised, fragments like lipopolysaccharide can pass into circulation and trigger systemic immune activation. That inflammation can activate microglia, the brain's immune cells, weaken the blood-brain barrier, and increase inflammatory signaling within neural tissue. The downstream effect is reduced synaptic integrity and impaired cognitive function. Another pathway involves the stress axis, where microbial byproducts influence hypothalamic pituitary adrenal signaling. Dysbiosis is linked to elevated stress hormones and reduced sensitivity to the body's stress-calming feedback, meaning stress responses stay activated longer.
Microbes themselves also produce or degrade neurotransmitters and their precursors, including GABA, glutamate, serotonin, dopamine, and norepinephrine. They generate signaling molecules like tryptophan derivatives and short-chain fatty acids that influence brain-derived neurotrophic factor expression. Dr. Mercola also notes that dysbiosis is associated with impaired insulin signaling, which affects brain energy supply, a factor that matters for both mood regulation and cognitive stability. The picture that emerges is not a single pathway, but an interconnected web of biological communication between gut and brain.
From those mechanisms, Dr. Mercola transitions into foundational strategies for supporting gut health. The first emphasis is lowering linoleic acid intake, the omega-6 polyunsaturated fat found heavily in industrial seed oils like soybean, corn, sunflower, safflower, and canola. Excess linoleic acid disrupts gut integrity by irritating the intestinal lining and promoting inflammatory conditions that allow dysbiosis to establish itself. The target is keeping total linoleic acid intake under 2 grams per day from all sources, and the recommended cooking fat replacements are grass-fed butter, ghee, or tallow.
The second strategy involves rebuilding the gut with appropriate carbohydrates. Dr. Mercola recommends sweet potatoes, carrots, squash, and cooked white rice as sources of fermentable fibers that feed beneficial bacteria. But he introduces an important caution called the fiber paradox. If the gut is already damaged, increasing fiber too quickly can backfire with bloating, cramping, or constipation. The recommendation is gradual introduction in small amounts, with most adults doing well around 250 grams of healthy carbohydrates per day once their digestive system has adapted.
Stress management, sleep optimization, and regular exercise complete the framework. Chronic stress alters the microbiome through hormonal signaling. Sustained stress hormones weaken gut barrier function, and the resulting microbial shifts feed back into brain function. Dr. Mercola recommends meditation and structured breath work to bring stress physiology toward a healthier baseline. For sleep, complete darkness with blackout curtains, limiting blue light at night, and reducing electromagnetic field sources. For movement, daily walking serves as a meaningful starting point for influencing stress hormones, metabolic stability, and inflammatory tone.
The research does not claim gut imbalance single-handedly causes bipolar disorder. What it demonstrates is that the microbiome can influence inflammation, dopamine signaling, synaptic structure, and cognitive function in brain regions directly involved in mood and decision-making. The practical takeaway applies broadly to anyone interested in supporting mental health through biological terrain. Reduce seed oil exposure, rebuild microbial diversity gradually, manage stress, optimize sleep, and move consistently. What is particularly encouraging about this research direction is the reversibility evidence. In the animal models, restoring a healthy microbiome partially recovered the structural and functional brain changes that dysbiosis had caused, which suggests the biological terrain is not permanently fixed once disrupted. That creates a meaningful window for intervention. For the complete study breakdown, including the specific data on dendritic spine density, dopamine circuit changes, microbial profiles that may help distinguish bipolar from major depression, and the full gut support framework. Visit Mercola.com and read the full article on gut bacteria and bipolar depression.
Thank you for listening to Dr. Mercola Cellular Wisdom. See you in the next episode.
Looking for trusted health wisdom you can hold in your hands? Discover the life-changing books of Dr. Mercola at joyhousepublishing.com. These titles bring decades of natural health research into practical steps for better energy, stronger immunity, and clearer thinking. Take charge of your health with guidance rooted in science and experience. Visit joyhousepublishing.com now and claim your bonuses while they are available.
Thanks for watching. Subscribe now and click the notification bell so you never miss an update. See you in the next video.